So today I would like to talk a little bit, okay, about this first part, in which we will talk about the tools used in the gene cloning procedure, okay, and so I will start with this one, okay, an introduction, okay, reintroduce to you guys about what is actually the recombinant DNA technology. Okay, so basically, when we talk about the recombinant DNA technology, we are focusing on a technology, a type of technology, in which this will be referring to a procedure which will recombine DNA molecules. Okay, maybe one DNA molecule from one organism is joined with another DNA molecule from another organism, another different species, and this will be producing a new type of DNA molecule, which we call as the recombinant DNA. Okay, why recombinant? Because it is a combination of two molecules of DNA from two different species. Okay, what is so important about this technology? Of course, this is one of the latest technology in the field of biology, in the field of biotechnology, and with this particular type of technology, scientists will be able to make many copies of a particular DNA segment or a particular gene, a particular type of gene. In other words, when we say that we are making many copies, we are actually cloning the DNA segment. Okay, so one of the important things related to this technology will be about the gene cloning, about how we make many copies of a particular gene. Okay, and we can also put a different type of DNA, a foreign DNA, into another cell, okay, of another organism. Okay, so these are just some of the importance lah, of this particular recombinant DNA technology. Okay, in the field of biotechnology, there will be so many things that scientists can do. Okay, but one of the things that can be done under the field of biotechnology is to make many copies of genes, or make many copies of certain DNA fragments, to clone or to amplify. Amplify means to make many copies. So this will involve the recombinant DNA technology, in which we combine two DNA molecules from two different organisms. Okay, and then we can proceed with the gene cloning procedure. Okay, another technique in order to clone certain gene or certain DNA fragment will be the PCR, the polymerase chain reaction. But for this matriculation syllabus, we will be focusing more on this gene cloning. Okay, we will be focusing on this part. For the PCR, you just have to know just a little bit. Maybe you can read a little bit about this from the internet. Okay, this is more high tech lah. And so we focus on this, in which you just need to know what will be the process, okay, of the gene cloning procedure, okay, involving these several steps. And in order to do this gene cloning process, we need to have some tools. Tools here will be referring to the things that scientists use in order to do this gene cloning procedure. Okay, so there will be 5 important tools that you need to know. Okay, so we will focus on the tools now. Okay, the first one will be the target DNA. The target DNA, which will be referring to the DNA molecule. Okay, DNA molecule, which is actually the chromosome lah, the chromosome found within the nucleus of a particular cell. Okay, and this target DNA is a special DNA molecule. Why? Because that is where we can find the gene of interest. Okay, the gene of interest will be the gene that we would like to study, the gene or the DNA fragment that we would like to clone. The gene or the DNA fragment that we would like to make into many copies. Okay, kita minat satu gene saja. Okay, so in this long DNA molecule, there might be many genes. Of course, there will be many genes in this one long DNA molecules. Okay, but we are interested on only one particular gene. Okay, and so this whole DNA molecule can be considered as the target DNA. Why? Because it contains our specific gene of interest. Macam ni lah. And this will be the long DNA molecule. This is the gene of interest for us to study. Okay, so that will be the target DNA, and usually we will use this particular target DNA in order to make many copies of our gene of interest, or maybe to produce a particular type of protein. Okay. The next tool will be the cloning vector. The cloning vector will be actually referring to a type of DNA molecule. Okay. And when we say that it is a vector, you must understand that a vector will be something that will be able to carry, it will be able to carry some other things. In this particular case, this cloning vector is a DNA molecule that can carry another DNA molecule, and this DNA molecule is considered as a foreign DNA, DNA asing. Maksudnya DNA daripada sumber lain. Okay. We can also say that this foreign DNA is the gene of interest. Okay, so a cloning vector will be the DNA molecule that can carry our gene of interest into a host cell, in which the host cell will later make this gene of interest into many copies. Okay, so you must understand. Okay, if you have watched the video, you will see that in order to make many copies of this particular gene of interest, we will need to put this gene of interest into a cell that will be able to reproduce rapidly. Okay, and when the cell reproduce rapidly, the gene of interest will be multiplied, will be made into many copies. So that cell will be known as the host cell, and we will talk about this later. This is also a tool. Okay, the important thing now is we will be talking about this cloning vector that will be carrying the gene of interest into the host cell. Okay, and one example of a cloning vector will be a plasmid. Okay, actually there are many types of cloning vector. There are many types. For example, the bacteriophage. Another example will be the cosmid. Okay, but we will need to highlight this particular one. Okay, the plasmid. Okay, what is a plasmid? A plasmid is actually a DNA molecule that can be found within a bacteria. Okay, so if this is a bacteria, this is the E. coli bacteria. Okay, this will be the chromosome, the main chromosome of the bacteria, the main DNA molecule of the bacteria. Okay, every bacteria should have this main chromosome lah. Okay, but some of the bacteria will be having another accessory DNA molecule. We call it as the accessory DNA molecule. Why accessory? Because some bacteria will be having this accessory DNA molecule, some will not. Okay, so this will be the accessory DNA molecule that we call as the plasmid. Okay, it is smaller than this main chromosome of the bacteria, and it is circular. Dia membulat like a ring. Okay, maybe there will be a few plasmid in one bacteria. Maybe there is just one plasmid in one bacteria, ataupun there might be no plasmid at all in a particular bacteria. Depends lah. Depends on the bacteria itself. Okay, so in this case, we can say that the plasmid, okay, can be obtained from the E. coli bacteria. Okay, and we can use this plasmid from the E. coli to become our DNA cloning vector, and that will be used in this recombinant DNA technology. Okay, and in order to use this plasmid, we need to remove, we need to take out this plasmid away from the E. coli bacteria, like this. Okay, this is very small. This is very small. We cannot even see this plasmid actually, using our naked eyes. Okay, and you need to know the characteristics of the plasmid. Okay, you need to know about this because this is one of the favorite questions in the exam or in the UPS. Okay, so you just look through the notes, okay, in which you are given these 4 characteristics about the plasmid. Okay, for example, the plasmid should be able to accept the foreign DNA or the gene of interest. The plasmid must be able to accept the gene of interest or accept the foreign DNA in multiple cloning site location of the plasmid that can accept this DNA from other organisms. Okay, and the plasmid should be able to replicate freely in the host cell, meaning that if we put this plasmid into a bacteria, this plasmid can replicate, meaning that this plasmid can from 1 can become 2 and so on lah. And this is because of the the origin of replication. And another characteristic that is important for the plasmid is that they will be having marker gene. So this is a plasmid, okay, and there is one gene over here that is very important, and we call it as the MPR gene. The MPR gene. The MPR gene is a gene that will give resistance to antibiotic. Okay, antibiotic is a substance that should kill the bacteria. However, if a bacteria has got the plasmid and the plasmid will be having this MPR gene, the bacteria will not be destroyed by the antibiotic, by the ampicillin. Okay, this MPR is specific for the resistance to ampicillin, a type of antibiotic. Okay, and usually this MPR gene, okay, can be considered as a marker gene, which is important later in the process of screening, okay, during gene cloning, during the process of gene cloning. Okay, and this is another characteristic lah. It is able to express or amplify the clone gene in under suitable condition. Okay, so this is one example of plasmid. Okay, one example of the cloning vector will be the plasmid, and actually there will be many types of plasmid. So this is a specific example of a plasmid. We call it as the PUC18. PUC18. Okay. For the third tool number 3 will be the restriction enzyme. Okay, the restriction enzyme, or sometimes we can also call it as the endonuclease. Okay, but we prefer you to use this term, the restriction enzyme. Okay, and from the word restriction, we should know that this involves cutting. Restriction means to cut, to potong, and so this is a type of enzyme that will cut or cleave, cut or cleave, potong, dua-dua sama maksud, to cut or to cleave a particular DNA molecule. Okay, so this restriction enzyme will be able to cut a particular DNA molecule at a specific restriction site. A specific restriction site. So normally this enzyme will recognize first. This enzyme will go to the DNA molecule and it will recognize. It will recognize a particular restriction site. This is where the enzyme will cut the DNA molecule. Dia akan cari dulu, mana ada restriction site, di situ dia akan potong. Okay, so how to identify a restriction site? Okay, normally the restriction site will be symmetrical, and the important thing is the restriction site will be palindromic. Okay, palindromic. What is the meaning of palindromic? Palindromic is a condition in which if we look at these two strands of DNA molecule, remember, one DNA molecule consists of 2 polynucleotide strands, okay, which are parallel to each other and complementary to each other, remember? Okay, so this palindromic sequence shows a sequence of bases of the DNA which will read the same GAA TTC, and for this strand over here we also read the sequence of bases the same GAA TTC, however in opposite direction. Okay, for this strand up here, we read the sequence from left to right, GAA TTC. For this strand down here, we also read it as GAA TTC, but in the opposite direction, from the right side to the left side. Okay, basically, when we read the sequence of the bases, normally the direction will be from 5' end to the 3' end. Okay, so palindromic here shows this condition in which I think you can notice that the reading will be the same, but in opposite direction, and that is palindromic. Okay, and we can see here that this involves a sequence of 6 bases, 6 nucleotide bases, 1, 2, 3, 4, 5, 6, and sometimes it can involve only 4 bases or maybe up to 8 nucleotide bases, but the common one will be this 6 sequence of bases lah. There are many types of restriction enzymes. What we need to know about these two examples, and between these two, this one is the most important one, the EcoR1. The way we pronounce it will be the EcoR1. Meanwhile, this other restriction enzyme is known as the SMA1. Okay, and basically all the restriction enzyme will cut the DNA molecule by cutting the phosphodiester bond between certain nucleotides of the DNA molecule. Okay, and this will produce either the sticky ends or the blunt ends. Okay, so what will be the sticky ends and blunt ends? Okay, we will use this example of EcoR1. Okay, in which the restriction enzyme known as the EcoR1 is actually an enzyme that can be obtained from a bacteria. Okay, and that bacteria will be the E. coli. Okay, so you must remember that EcoR1, okay, the EcoR1 enzyme can be obtained from a bacteria known as E. coli. Okay, so this EcoR1 will cut the DNA whenever this enzyme recognize a particular palindromic sequence, and the sequence will be GAA-TTC. You must remember this sequence, special for the EcoR1. Okay, GAA-TTC, if we read it from the 5' end to the 3' end direction. Okay, and this EcoR1 will cut the DNA molecule, okay, in this particular way, in which the EcoR1 will make staggered cut. Staggered cut means the cutting is not continuous. Okay, dia macam tersekat-sekat. Itu maksudnya staggered cut. Okay, pemotongan yang tersekat-sekat. Di mana we can see in this diagram, okay, this will be the palindrome, the palindrome GAA-TTC that will be recognized by the EcoR1, and the EcoR1 will cut, okay, between the first base and the second base, between the G and the A. Okay, the cut, okay, somewhere over here, macam ni. Okay, and the same goes on this other strand. The EcoR1 will also cut between the first base and the second base. Kita anggap ni satu the first base, because we read the sequence from this end, the 5' end. So we consider this as the first base of the palindrome lah. Okay, so the EcoR1 will cut between the G and the A, between the first base and the second base. Okay, so they cut over here, they cut over here. So dia macam dia tak berlaku secara continuous. Dia betul sini pula. Okay, and as a result, when the phosphodiester bond over here is destroyed, the same goes for this phosphodiester bond over here is destroyed. This will separate the DNA molecule to become like this. Okay, so this part over here, okay, this part over here will become this thing. Okay, meanwhile this second part is now separated separated, and it will become this end of the DNA molecule. And we can see that this particular strand is protruding, dia tertonjol keluar. So this is known as the sticky end, this part over here. Okay, the same goes on this side. Okay, this part of the DNA strand which is protruding will be producing the sticky end, another sticky end. Okay. Now what about this other restriction enzyme, the SMA1? The SMA1 is another type of restriction enzyme that can be obtained from another type of bacteria. Okay, another type of bacteria, the SMA1. Okay, the bacteria will be, I cannot really remember the name. I think it will be Serratia. Okay, Serratia. I'm not very sure about the spelling. Spelling of the species name, Serratia marcescens. I'm not sure about this spelling. Okay, so I just put it as, if I'm not sure about the full scientific name, I will write it as Serratia species. Okay, to be on the safe side. You can read about this from the internet. Serratia marcescens, I think. Okay, but I'm not sure about the spelling. Okay, the more important thing will be the E. coli that is obtained from the E. coli. That is important for you to remember. This is just something extra lah, Serratia ni. Anyway, SMA1 will be another type of resistance enzyme that will cut a DNA molecule whenever this enzyme recognize another type of palindrome. Okay, and the sequence of the palindrome will be CCC GGG. If we read from the 5' end to the 3' end direction. Okay, the difference between the SMA1 and the EcoR1 is that this SMA1 will do a straight cut. A straight cut in which we can see the cutting will be straight like this in the middle of the palindrome. The palindrome is also consisting of 6 bases of DNA. Okay, 1, 2, 3, 4, 5, 6, and the SMA1 will cut in the middle of the palindrome. Okay, in the middle between the base number 3 and the base number 4, between the C and the G. Okay, and so when this SMA1 cut the DNA molecule like this, straight cut, okay, this will produce the blunt ends, in which, as you can see here, there will be no strand that will be protruding. Okay, semuanya tak tertonjol, semuanya sama je kan, straight aja dia punya hujung dia, hujung dia straight aja. So this is known as the blunt ends, hujung tumpul in Malay, hujung tumpul. So there will be blunt end over here, another blunt end over here. Okay, and actually this one is more important actually, sticky end. In the process of gene cloning procedure, normally sticky ends will be more important, okay, more useful. Okay, anyway, we still need to know lah about the production of the blunt ends. Okay, tool number 4 will be the modifying enzyme. Okay, modifying enzyme means an enzyme that can modify, that can change the condition of the DNA. Okay, and there are 2 examples of modifying enzymes that are mentioned in the syllabus. One will be the DNA ligase. Okay, and another one will be the Taq polymerase. Taq polymerase enzyme is a type of enzyme that is important in the PCR. We don't need to learn in detail about this PCR. Okay, we just mentioned just about this just now, and you can read on your own lah about the polymerase chain reaction, which involve this enzyme Taq polymerase. In the process of gene cloning, we will use this DNA ligase. So it is more important for you to discuss about this particular modifying enzyme, the DNA ligase, in which this is the same type of enzyme that you have used before this in the topic number 6, when you learn about the replication of the DNA. Remember, when we want to join the Okazaki fragments, that will also involve the DNA ligase. In this story of the gene cloning procedure, the DNA ligase will be used to join the gene of interest or the foreign DNA with the cloning vector, and this later will form the recombinant DNA molecule. Okay, so dia akan sambungkan, lagi ni akan sambungkan dua DNA yang berbeza menjadi satu DNA baru yang namanya adalah the recombinant DNA. Tool number 5. Okay, tool number 5 will be the host cell, sel perumah. So this is actually referring to a type of organism that will be able to reproduce rapidly. Okay, and usually we will be using the bacteria as the host cell for this gene cloning procedure. Okay, why? Because the bacteria is a type of organism that can reproduce rapidly. Okay, from one bacterium, it will perform binary fission to produce 2 bacteria. From 2, it will become 4 bacteria. From 4, it will become 8 bacteria. From 8, it will become 16 bacteria. From 16, it will become use your own calculator, please. Okay, and the important thing is we will use this host cell, okay, in which this bacteria, this organism, will receive the recombinant DNA in order to allow the recombinant DNA to form many copies. In which we can say that we use this host cell to clone the recombinant DNA, so that this recombinant DNA, so that our target DNA, so that our gene of interest will be forming many copies. Okay, so the example of the host cell that we normally use will be this bacteria E. coli. So if you notice, the bacteria E. coli is very, very popular in this procedure of gene cloning. Okay, because this bacteria has got many purpose. Okay, number one, just now we say that the E. coli can be the source of the cloning vector that we call as the plasmid. Okay, that is number one. E. coli is also useful in the gene cloning procedure. Why? Because the E. coli can give us the restriction enzyme known as EcoR1. And the third importance of the E. coli is that they can become the host cell for this cloning procedure. So this E. coli is very, very useful in the field of biotechnology actually. Okay, and please take note, everybody, about the characteristic of the host cell. This is another thing that you need to remember. Just now we talk about the characteristic of the cloning vector of the plasmid. Now you also need to know about the characteristic of the host cell. Okay, for example, of course, the host cell must be able to receive the recombinant DNA. Okay, and this will involve the process known as transformation. Okay, whenever a DNA molecule gets into the bacteria, we say that transformation occurs. Okay, and the host cell must be able to maintain the structure of the recombinant DNA. Okay, meaning that the host cell will not disrupt the original structure of the recombinant DNA. So once the recombinant DNA gets into the bacteria will just maintain the structure from one generation to another generation to another generation. Okay, and the host cell must also be able to amplify the recombinant DNA. Amplify means it must be able to make many copies of the recombinant DNA, and the host cell will also be able to express the gene of interest. Okay, meaning that the host cell will be able to produce a particular type of protein special for the gene of interest. Okay, and so those will be the tools that are important for the procedure of gene cloning. And remember, there are 5 tools: the target DNA, the cloning vector, the restriction enzyme, the modifying enzyme, and the host cell. Okay, and so after we have learned about the tools, okay, of course, we need to continue with the gene cloning procedure. And I just want to show you about this diagram. Okay, you can read on your own about the steps in this gene cloning procedure, and I've also tried to show the important steps through my video. Okay, in which we will start with the first tool. On the right, we will be focusing on the first tool, that will be the target DNA. That will be having the gene of interest. Okay, so the first thing that we need to do is we need to isolate. Isolate means we remove, we take out, we remove the target DNA from the cell. We can call this cell as the target cell, or we can call this cell as the donor cell. Okay, the important thing is inside the cell will be the nucleus, and inside the nucleus there will be the chromosome, a particular chromosome that we will consider as the target DNA when it contains the gene of interest. So this is something that you have done in your experiment number 6, and also your practical test. Remember what you did for your practical test? You isolated the DNA from a fruit cell. Okay, so this is the same technique actually, in which we isolate, we remove the DNA from the cell, and this will be the target DNA that has been removed, that has been isolated. Okay, and in this long target DNA there will be our gene of interest. Okay, that is step number 1, isolation. Isolation. And we also need to do another isolation, and this one will be referring to the isolation of the DNA cloning vector. Also the isolation of DNA, but this one is specifically referring to the isolation of the DNA cloning vector, or the isolation of the plasmid, in which I've told you just now that the plasmid is something that can be found in a bacteria. Okay, and so we also need to isolate, we also need to remove the plasmid out from the bacteria, from this E. coli. So this is the plasmid. Okay, step number 1, isolation. Okay, the removal of target DNA and also the removal of the cloning vector. Okay, step number 2 will be cut or cleave. Okay, in which we need to have this gene of interest. Okay, so we need to cut this long DNA molecule, and we can cut using the tool number 3. Okay, this is tool number 1, the target DNA. This will be tool number 2, the cloning vector. Now we are using tool number 3, the restriction enzyme, in which we can use maybe the EcoR1. Maybe we can use the EcoR1 that can also be obtained from another different E. coli. Okay, and we use the EcoR1 to cut or to cleave this DNA molecule, and we will be able to obtain these fragments, fragments known as the restriction fragments. Restriction fragments, and one of the restriction fragments will be having our gene of interest yang color merah ni. So ada banyak kepingan DNA. Okay, those are known as the restriction fragments. Tapi salah satu daripada fragment tersebut ada kita punya gene of interest yang ni lah. Okay, and we also need to cut the plasmid. We also need to cut the plasmid, and so we need to know that we need to insert this gene of interest into the plasmid. Kita nak selitkan, kita nak sisitkan, so kita kena cut jugalah. We need to cut this plasmid using the same, remember, using the same type of restriction enzyme. So if you have used EcoR1 to cut the target DNA, you will need to use the same EcoR1, the same type of EcoR1 to cut this plasmid over here. This is to produce the complementary sticky ends. So that sticky end yang ada dekat gene of interest ni, dia akan padan, dia akan ngam dengan sticky end yang ada pada plasmid ni nanti. Okay, so after cutting, the second step is cutting the target DNA and cutting the plasmid. It will be continued with step number 3, insertion. insertion, in which we just insert this gene of interest, okay, into the plasmid, macam ni. Okay. However, if you notice, the gene of interest, okay, is not joined properly to the plasmid yet. Okay. So you must remember that these two are DNA molecules, but of different sources lah. This DNA molecule maybe comes from one type of cell. Meanwhile, this plasmid is also a DNA molecule that comes from a bacteria. Okay, but we can join these two DNA molecules together. How? By using tool number 4, the DNA ligase, a type of modifying enzyme. So by using this DNA ligase, we have joined this gene of interest together with the plasmid, and now terbentuklah. This will form the recombinant DNA, or the recombinant plasmid. Okay. Okay, you can call this whole thing, this whole structure, as the recombinant DNA or the recombinant plasmid. Okay, kenapa recombinant? Because it is a combination of two DNA molecules from two different sources. Satu datang daripada sel apa-apa entah, satu lagi datang daripada bakteria. So kita gabungkan dua DNA molekul bersama, dan jadilah satu DNA recombinant. Okay, and then we continue with step number 4. Step number 4 will be transformation, in which we just put this recombinant DNA into a host cell. So this will be our last tool. The last tool will be the host cell. We can use the E. coli. We can use the E. coli, and we put the recombinant DNA into this E. coli. So transformation has been done, and we call this bacteria as the recombinant bacterium, or we can also call it as the transform bacteria, transform bacterium, bacterium. Okay, maksudnya bakteria yang telah pun menerima satu DNA lain, transform bacteria. And after that, we continue with the process of amplification. Okay, amplification will be the step in which we will just let the bacteria to reproduce. Okay, so you can see from one bacteria, from one bacterium, this will produce many other bacteria, and at the same time, the gene of interest, this red colored thing, will also be made into many copies. Okay, and usually we need to identify which are the bacteria that really carrying our gene of interest. So we need to do some screening. Okay, this will be the last step, the screening process, or you can also specifically mention the word blue-white screening. Okay, so normally we use a dash for the word blue-white. We don't use a slash, we use a dash macam ni. Blue dash white screening. So betulkan if you see somewhere in your notes ada guna slash ni, betulkanlah. Saya punya rakan-rakan memberitahu ini yang terbaru lah. Kalau boleh kita gunakan dash bukannya slash. So if you notice my video pun ada tergunakan slash, pasal tu dah lama kan. So maafkan saya. You can change the slash into a dash. Okay, so using this blue white screening, we will be able to identify which are the bacteria which really carrying our gene of interest. Okay, so this will be the step that you can read on your own. Okay, and this will be the diagram that shows about the blue-white screening procedure. It is because of the plasmid. The plasmid is actually having this marker gene, the MPR gene, and also the LEGZ gene. Okay, remember the LEGZ gene? We have talked about this LEGZ gene when we talk about the leg operon in topic number 6. Okay, in which we know that the LEGZ gene will code for the production of the beta-galactosidase enzyme. Okay, and so if we put a gene of interest into the plasmid, normally the gene of interest will be somewhere within the leg Z gene. Okay, so once the gene of interest is inserted, okay, within the leg Z gene, the leg Z gene will not be able to function. The leg Z gene will not be able to code for the enzyme beta galactosidase. Okay, and that is why when we do this blue-white screening, okay, any bacteria that do not have any plasmid at all, do not have any this plasmid, will not grow in the petri dish. Why? Because of the ampicillin. Ampicillin is the antibiotic that will kill any bacteria without the plasmid. Why? Because the bacteria without plasmid will not be having this gene. Okay, a bacteria that has got this plasmid with the MPR gene will be able to resist the effect of the ampicillin. Okay, so kalau bacteria ni dia tak ada langsung plasmid, okay, maksudnya dia tak ada gene ini, maka dia akan matilah. Kita tak nak pun bacteria ni. We don't want this type of bacteria. Okay. Meanwhile, some of the bacteria will not be carrying any of the gene of interest. Tak ada pun. Okay. This bacteria has got a plasmid, but the plasmid is original, the original plasmid, or we can say that this is the non-recombinant plasmid. The non-recombinant plasmid. Okay, this is not we want. Okay, tapi dia masuk juga dalam bakteria. We don't want this one. And normally, this type of bacteria will be producing blue colonies. Titik-titik yang ada kita nampak dekat agar-agar ni, ini adalah koloni-koloni bakteria yang sebegini, yang tidak ada recombinant DNA. Dia ada non-recombinant plasmid saja. So we don't want this blue coloured colonies. Okay. What we really want will be this type of bacteria, a bacteria that has got a recombinant plasmid. Di mana kita nampak sini plasmid dia ada gene of interest. Okay. Tapi oleh kerana dia ada gene of interest terselit di dalam leg Z gene dia tidak akan berfungsi, maka tidak akan ada penghasilan enzim beta galactosidase, and that is why dia akan menghasilkan tumpuk-tumpuk putih. Dia akan menghasilkan white colonies, bukannya blue tapi white colonies. So whenever we see white colonies in the petri dish, that will be the type of bacteria that we really want. Okay, kerana dia kalau dia punya leg Z masih lagi berfungsi, leg Z dia akan menghasilkan beta galactosidase enzyme yang akan bertindak dengan X-gel, dan dia akan menukar X-gel kepada warna putih, kepada warna biru. Okay. Tapi kalau dia punya leg Z gene dah tidak berfungsi akibat ada terselit gene of interest, then the X-gel will not be hydrolyzed by the beta galactosidase, and the X-gel will remain as white substance. So kita nampak akan ada tumpuk-tumpuk koloni putih. Okay, you can read more about this blue-white screening from the internet. And so that is all about the gene cloning procedure. After this, you just need to know about the application of the recombinant DNA technology, in which there are many things that can be done actually using this recombinant DNA technology. Okay, in agriculture, in the field of environment, in the field of food production. But according to our syllabus, we need to know about this insulin production. How to produce insulin using the recombinant DNA technology. So basically the procedure is about the same. Okay, it's about the same. Perbezaannya adalah, instead of removing the target DNA, okay, you can see here that we remove the mRNA. Itu perbezaan dia. Okay, I repeat, instead of isolating the target DNA, over here we isolate the mRNA. Okay, and later you will be able to see that we will produce the cDNA, the complementary DNA. So this is what is different. This is what different between this procedure in order to produce the insulin with the gene cloning procedure. Okay, so I will end my lecture, okay, here, and I will give you another video focusing on the application of recombinant DNA technology, specifically focusing on the production of insulin. Okay.